Thank you. So for something completely different, the sun is shining at last in Cambridge, and I think most of us would have experienced the pain and discomfort of sunburn at some point in our lives, and so it's pretty apt for me to be able to tell you about how to use sunburn, or at least the UV radiation from the sun's rays, as a translational research tool. But first, a quick disclaimer. This presentation includes research conducted while I was employed by Pfizer and does not present the results or represent the views of my current employer, which is MedImmune. But I am a steering committee member of the European Union Innovative Medicines Initiative, European Consortium. I previously represented Pfizer and do now represent AstraZeneca in this role. So I'm going to tell you about the development and validation of the UV pain model, but more importantly about the application to translational pain research. Firstly, for the identification of novel targets, and then secondly, for proof of mechanism. By proof of mechanism, I mean confidence, high confidence that we're hitting, that the molecule is hitting the target and affecting a relevant pain mechanism. I have quite a bit of data to share. However, I recognize that this is a case study, and so I will have to gloss over some of the details and hopefully point out some lessons learned and critical success factors for successful clinical, preclinical translation working in two contexts. First, in an open innovation, public-private type consortium that involved academia, Pfizer, AstraZeneca, and other pharma academic partners, and then secondly, in-house validation for internal proof of mechanism decision-making within one company, that's Pfizer. So in the human model, we irradiate the skin with 3 times the minimum erythema dose. You can do that on the thigh or on the forearm, and one could look at various pain endpoints, pain-related endpoints that tell us something different about pain mechanisms. So we could look at heat pain, perception and tolerance thresholds, use a thermode, ramp the temperature up from skin temperature up to 50 degrees, avoiding any burn in the subject. We could look at mechanical pain thresholds and stimulus response using a von Freyhairs or mechanical needles. One could look at the area of secondary hyperalgesia beyond the primary erythema area, and this would tell us something about central hypersensitivity. And then finally, one could look at dynamic allodynia using a brush or cotton wool. So this is pain generated with a normally non-painful stimulus. The UVB radiation promotes long-lasting erythema that we know about, but also significant sensory changes in human skin. So the heat pain threshold goes down with increasing doses of UVB. Mechanical pain thresholds also decrease with increasing dose, and you see the dose responses here from control to 3 times the minimum erythema dose, which I said was the dose that we apply to the human. The UVB model of inflammatory pain is mechanistically plausible. So that's the first thing that we have to be convinced about. I haven't got time to go through all the evidence, but certainly UV radiation has been shown to release a wide range of inflammatory mediators known to be key players in inflammatory pain. There is also pharmacological evidence. Anti-inflammatory cytokines and sequestration of nerve growth factor, NGF, blocks the hyperalgesia, and well-known gold standard analgesics such as non-steroidal anti-inflammatory drugs, opioid analgesics, and COX-2 inhibitors have been shown to be active preclinically and clinically. So let me tell you about the EuroPain Consortium. This is a public-private consortium for translational research, which is part of the European IMI, Innovative Medicines Initiative. All the partners are are shown here. The logos for all the partners are shown here. Involves pharma partners which are active in pain research in Europe, academic partners from the London Pain Consortium, the Danish Pain Research Network, the German Pain Research Network, and a Spanish SME. So the aim is to collaborate, partner in an open innovation format to increase our understanding of pain, chronic pain mechanisms, and our big knowledge gaps here, to facilitate the development of novel analgesic drugs, and then finally to improve the management of chronic pain in patients in Europe and beyond. It's funded to the tune of 6 million euros over a 5-year period with, from EU funds, but importantly also 11.5 million euros worth of pharma in-kind contributions. So it's a significant investment. So it started in 2009, so we're in the 4th year of the consortium. And so I'll tell you about how this was applied for the UVB model was applied for the identification of novel pain targets. Now, traditionally, the pharma industry has been highly secretive about targets. We think there's IP involved, and there's a closely guarded secret. But the fact is, we get target selection wrong most of the time, and I don't think that's a controversial statement anymore. And this really is an attempt to be much more open and work collaboratively in target identification, and let the competition begin in terms of designing the best molecules. So the work here was conducted in, essentially in Steve McMahon's lab at King's College London, but although the work is now published, all of the data, the ongoing research was always shared very openly throughout the project, and it's still ongoing, obviously. So Steve McMahon's group basically irradiated both rat and man with a UVB radiation, and they took skin biopsies, punch biopsies from the rat hind paw and human forearm. They then looked at gene expression using low-density PCR arrays. Now, we are at the Sanger Institute, and I do have to tell you that we've moved on to next-generation sequencing. But the data I'm going to show you is just simply based on these PCR arrays. So this is sort of partially hypothesis-driven in that a number of genes known to be, or at least putative inflammatory mediators were profiled. Most of these genes were expressed in both human and rat. The UVB irradiation induced gene expression changes that are positively correlated in both between human and rat. And the top candidate from this profiling was the chemokine CXCL5 with a greater than 60-fold change in human. So that's a target worth further investigation, and indeed we went on to do that. So further work, detailed work on time course showed that the CXCL5 expression matches the time course of UVB mechanical sensitization. You can see the peak of mechanical sensitization with the lowest withdrawal threshold is about 48 hours, and here we have the greatest fold change in transcript expression. And when we looked, when the group looked at the protein levels, they also found that the expression was significantly increased after UVB irradiation, but was undetectable in control. They then went on to inject CXCL5 into the rat hind paw. So the local injection caused a dose-dependent mechanical pain-related hypersensitivity, but not thermal hypersensitivity. Further, they injected a neutralizing antibody against CXCL5 and showed that the local chemokine neutralization attenuated the mechanical sensitization after UVB. So So this is an interesting target, and I can certainly tell you that it's undergoing further target evaluation and validation. So next I'm going to tell you about a validation of the model for internal proof of mechanism decision making in the context of analgesic drug development. So this is work conducted by Pfizer. Now, it's all very well to move drugs forward when we've got a positive result, with any model or any clinical study. But the fact remains that most of the clinical trials in neurosciences and in pain research end up with a negative result. And often we just don't know why. We don't know, we're not confident that we've really hit the target and done good science, actually showed that we've tested a scientific hypothesis. So it's vital that before one uses any model, and a significant publication bias in the literature, that before we use any particular model for decision making, that we have some sort of hands-on experience and have optimized the experimental conditions and the endpoints for the purpose of decision making, such that we could walk away with confidence if we get a negative result and are able to stop development and waste further development and funds. So this is just one piece of evidence here. A series of Pfizer studies were conducted to optimize the experimental parameters, the endpoints, and to demonstrate reproducibility and pharmacological sensitivity. So this is benchmarking the opioid oxycodone in controlled release formulation and the non-steroidal anti-inflammatory drug ibuprofen. So, and the work showed that the most reliable endpoint in terms of reproducibility, sensitivity, is the heat pain tolerance threshold. So this plot here shows that the difference from placebo and oxycodone increasing the heat pain tolerance threshold over 2 to 4 hours significantly. Ibuprofen also significantly increased the heat pain tolerance threshold at 2 hours, and then the effect drops off. And this is entirely consistent with the known pharmacokinetics of these drugs. Then the Pfizer team went on to develop the preclinical model for preclinical proof of mechanism. And as we've learned in conjunction with the work done in the consortium, irradiation of the hind paw also showed a significant thermal hypersensitivity, and the Pfizer team were able to do translational pharmacology in the rat, demonstrated that ibuprofen and oxycodone are positive in this model as in the human model, although the endpoints used are obviously clearly different. One thing that we learned was that the exposures required in the preclinical model were higher than that of the human model. So in the interest of time, I'll just show you this summary slide that shows the various criteria that was applied for fit-for-purpose validation. So I've shown you that this is a mechanistically plausible model. It was reproducible in terms of thermomechanical hypersensitivity, but not for secondary hyperalgesia. There's just two variables for this. There was pharmacological sensitivity to multiple mechanisms, giving confidence to test pharmacology. However, the dynamic range was too shallow and for detailed PK/PD modeling. So we know about that. The model is translatable, and importantly, I just want to make the point here, it was a pretty robust model. We were able to reproduce these results in different laboratories using different observers, different methods of erythema assessment, and multiple study sites. So in summary then, I hope I've shown you that the human experimental pain models are potentially useful for translational pain research. The UVB model has cross-species translatability and has utility for both target identification and decision-making for proof of mechanism decisions. So hopefully I've shown you two successful examples of good public-private consortia innovation and internal validation for robust decision-making within a company. And finally, I'd just like to acknowledge my former colleagues, and consortium partners who conducted the work. Thank you for your attention.
Thanks very much. We'll skip through a new modelling page. Any questions? Yeah. Wonderful. Thank you.
There's quite a big difference between placebo and no treatment when it comes to pain. What do you mean by placebo with a rat?
So in a rat, one could only do vehicle control, but the important thing is that these studies have to be done double-blind. Okay, so you know that it's still, you know, these results, you know, preclinical results are potentially susceptible to observer bias, but the important thing is vehicle control, i.e., you know, and double-blind. The observer does not know what drug or vehicle has been given.
But I mean, are you using as a... I don't understand. Do you mean there's somebody who has no treatment, somebody who thinks they've had treatment, somebody who...
So the drug or vehicle administration is done by somebody else, from the one doing the experiment and from the one doing the readings. So the observer never knows whether a placebo or a vehicle has been given or a drug has been given. I mean, true understanding of a real placebo effect in experimental animals is a different issue.